Okay. So I kind of want to introduce this topic to you guys and kind of think about this analogy a little bit as we go through some of the other examples. So just bear with me for a moment. But I don't know how many of you are familiar with a hydroelectric dam, pretty much how it works. Okay, but you're stacking up water pretty much behind a wall, and that water is going to be allowed to move through a specific channel, a very specific channel. In this case, this is the channel the water is coming out and has a huge amount of kinetic energy as a result of that. And this is pretty much how a hydroelectric dam is working. Okay, let's cut away the wall and look at the inner workings of this. Okay, so behind the wall, there's going to be a lot of water. How I'd like you to view that is on a membrane where we have particles that are stacked up, whether we're talking about water or ions or charges or anything really, that represents potential energy. So anytime you have particles stacked up on one side of a membrane, you can create a gradient that should represent potential energy to us. That's stored energy. And you can do work with that, okay? And once we start allowing those particles to pass through a channel or a gate, it's going to transfer our potential energy into a kinetic state. Okay, so as you see here, we're letting the water move through a turbine in a hydroelectric dam, and we're able to spin electromagnets or some other mechanism to be able to generate the electricity. In a cell, it's going to work pretty darn similar. Okay, so we're going to allow these particles to move through a very specific channel, and we're going to use that energy to get work done within cells. Okay, so I hope that analogy will resonate here as we go through some of these other slides. All right, so let's get back to me jumping in the river. All right, so what would happen if we go out behind the school and I said, hey, let's swim up to Springfield? All right, so I'm struggling right now. That's a lot of work to go north in that river, right? I'm moving against those particles of water. I have to fight the kinetic energy, and obviously, there's going to be a huge expenditure of energy there. All right, so anytime that happens in a cell, we're going to call that active transport. Okay, it's an active process. If you stopped at any moment in time, the particle would just go in the opposite direction. And I hope that is clear to everybody. Okay, so let's look at our first example of active transport. It's arguably one of the more important examples that we see in life. So a mitochondria and a chloroplast is going to be the site of this kind of activity here, but or a bacterial cell. But typically what's going to end up happening with a proton gradient is these dots represent protons, positive charges. Pretty much what we're talking about is a hydrogen ion, H plus, or a proton, which is just a single proton. These things are going to get stacked up on one side of a membrane. So if you recall from what I just tried to explain to you guys, this represents potential energy. This is energy that's enabled to do work. These particles do not want to be here, but because they're charged, they're not going to be able to pass directly through the phospholipids. They have only one option, and that's to go through a channel. Okay, so the fact that we can control their movement enables us to put a huge amount of potential energy away. The problem is establishing this state, because these things don't want to be here, right? I mean, it's hard to keep the water behind the dam. I mean, it's not the easiest thing in the world, and we have to move particles in this direction. Okay, so if we were just to open up any one of these transmembrane proteins, integral proteins, these protons would just flood through here, and I hope everybody would visualize that these protons, would just flood through this this protein. Okay, so the protons want to move in this direction, but they're not able to. All right, so the way this works pretty much in our bodies, without getting into too much details, is we're going to use these electron carriers to provide some energy, okay, through oxidation reduction reactions. They're going to be able to pass electrons, shuttle them from one protein complex to the next. And what we're going to see in these transmembrane proteins is that little bit of energy that is going to be released from the transfer of these electrons is going to be enough for us to alter this protein in a way that's going to enable us to push protons against the gradient. So this is how we can establish this huge potential energy force. Okay, is by continually passing electrons through this, which we call the electron transport chain. And if we stop that input of energy, this whole process would stop. We would not be able to continue to stack water behind that dam. Eventually, all of that potential energy would be turned into kinetic energy when we enable that to come through a molecule called ATP synthase, which we'll learn about later. For right now, I just really want everybody to understand that pushing things from low concentration to a higher concentration is called active transport, and this is one of the best examples of that in our bodies is something called a proton pump. Okay. The next example of active transport that I want to show everybody is this process called endocytosis. And endocytosis really is when you have a large food item. In this case, I used a chicken leg. Looks pretty good, right? But the cell wants to take this food item in. So what's going to happen here is that food item is actually going to start encroaching into the cell membrane. It's going to create this cavity where that food item will be packaged in a vesicle, and the membrane is going to move inward to pinch off a sealed food vesicle. And that food vesicle, hopefully everybody's thinking about this at this point in the course, would meet up with a lysosome or a series of lysosomes, and that food item will be digested and the parts used for cellular processing. Okay, so to break down this word, it's engulfing stuff inward. Okay, so taking things inside of the cell, large things, endocytosis. Okay, and there's two specific types of endocytosis. There's pinocytosis, specifically referencing the uptake of large quantities of fluid. So it could be like an oil. It also could be like an aqueous type solution. And then there's phagocytosis, which cells taken in, like we just showed you, a large solid. So those are the different types of endocytosis. And in nature, one of the best examples I can give you is this. Okay, so we have a big white blood cell here. And over here we have our small little bacteria. Okay, and you know, if this white blood cell is just cruising, all of a sudden it encounters this foreign bacteria. Bacteria is just chilling, minding its own business. What's going to end up happening is that white blood cell is going to absolutely engulf and take that in. Okay, this last process here that I want to explain to you in terms of a specific type of endocytosis is something called receptor-mediated endocytosis. So what happens is almost a very similar to what we showed you before. We have a part of the cell membrane, and out here we have receptors. These represent ligands, or the particles that are meant to be taken up into the cell. So these are receptors, and these are providing an initiation signal. So either these are the item that need to be taken up, or the concentration of these or the presence of these would trigger the formation of a vesicle. And we'll show you how that works. Okay, so what's going to happen is these will eventually bind with the surface receptor, and if enough of them bind to the surface receptor, we're going to have the formation of a vesicle. And so this is, again, you have a surface receptor that's going to mediate this process of forming a vesicle and creating that endocytosis. Okay, so last thing we're going to cover today, we have waste. Okay, and anytime we have waste that we want to have eliminated from the cell, what's going to end up happening is exactly the opposite of what we just saw. It's called exocytosis. Okay, so here we have a waste vesicle. This waste vesicle is going to fuse with the cell membrane. The phospholipids will just combine with what is the current membrane and expel the particles. And that's how cells eliminate large quantities of waste in the process called exocytosis. This also can be used to eliminate large quantities of product that is produced by the cell. But hopefully you got the gist, okay? And these processes absolutely require input of energy. And that's it for today. So hopefully everybody's um, picked up everything that we had to explain. Have a good night.